Good afternoon, everyone, and I really thank the organizers for considering me as one of the speakers, and it's a great moment and a proudish feeling to be back in CCMB because it's in 99, February 9, I came. It's almost a silver jubilee year. Came for the interview, and so it's a great feeling to be back here. And unlike Subhashini, I could not make any slides that explain, but I can verbally explain my stay and life in CCMB. So when I joined, I was given a project of growth enhancing or enhancing the growth rate of Indian major carp. So that was the project that was given to me, and at that time I did not know the risk involved. So just like any other beginner researcher, I jumped and grabbed, and then I realized the troubles involved with that, because Indian major carp cannot be handled in the lab. It needs river water, and there is no rivers in Hyderabad. And fish, its breeding time is 3 years. And so if I want to have a transgenic line in the second year of my PhD, I should have the transgenic line and delivered into. And there is no strategy standardized for transgenic line, developing transgenic line. So you have to make the road, walk on it, and pass the race. So it was a little difficult for me, but since you have no other option, you have to jump into it. So thankfully in 2 years I could make the auto-transgene construct. That means there is no viral element or no exogenous element is used. It has to be everything taken from the not even plasmid backbone is allowed. So we could develop a sperm electroporation strategy. So the fish will breed only in coastal Andhra. So we used to go to Krishna and Godavari rivers, and it will breed only at night, even after hormonal induction, maybe night 2 o'clock, 3 o'clock. So you have to be watching inside a hapa. Fish will be jumping, and that time it will be raining, and rain is not welcome, because if you collect the sperm or egg, it should not come in contact with water. If it is so, you can't do in vitro fertilization. So I used to go with umbrella, carry tissue paper, and fish will be around 3 kilo size. It will be wriggling. You have to wrap its eyes, and one fellow will be holding the fish, other fellow will be taking the eggs and collecting the sperm, and then you have to do sperm electroporation and then fertilize it. And end of the day, many a times it happens, fish will not survive, embryos will not survive, they all will die, because we are growing them in our own inbuilt hatchery, because they need everything natural, because it was one of the very finicky fish, Indian major carp. My species, what I chose was Labia rohita, but thankfully some luck was on my side, and in 2001 I could generate a transgenic line, and of the hundreds I could manage to survive only 5 of them, okay, and they are growing in our same pisciculture laboratory, which you can see adjacent to the auditorium what we have, and we are growing them in glass aquarium. And 2001 they are born, 2002 passed, 2003 passed, 2004 passed. I am on the 5th year of my PhD also. So we took that. Thankfully those fishes did not die, and they are transgenic, I know, because I have screened them, and we have to take them again with big syntax tank. We have to take those fishes, because they have grown little big also, because they usually grow very big. They can, in field it can grow up to 40 kilo size. So since our glass aquarium was small, it did not grow that big. So we had to go and put them, and finally in 2004 we could show there is transgene integration, and I confirmed by doing inverse PCR also, and that gave some way for completion of my thesis, and that is what my proposed thesis was. And of course various other experiments are there, but this was the main. After that, publication was a big hurdle, because due to, you can guess it, I will not go into the details, but as you can think about any boss and PI relation, so somehow I could manage to get one publication, and with that uncorrected proof I am applying for labs, and I applied around 25 labs, and thankfully nobody replies, and I can guess it why, and eventually one faculty from Vanderbilt University replied. I will not name it, it's not the right thing to say. And after a telephonic interview, no reply again. Then after 1 week I asked her, what about that? Then she replied a very philosophic email, and I have to dissect out what she meant, some sentences, eventually I could guess it that, it is a no from her side. And I said, okay. And again continued the applying, and thankfully from University of Michigan, Daniel Goldman, he did not interview me, he did not do any kind of interview. He said, Rajesh, can you come? He sent me some papers before that, and he asked, what do you want to work on? So based on my little knowledge on retina regeneration, I asked him, it would be this angle, it is better to explore. He said, oh, that is fantastic, because I have proposed an R01 grant. If I get that grant, you come. Thankfully, so this is 2 years past. After defending my thesis, I am here in CCMB for 2 more years as postdoc in the same lab. I am developing other, DNA vaccine construct for, white spot disease in prawns, etc. That's what I have been working on. I forgot to mention, I did my PhD with Dr. Kshitish Majumdar, who is no more. He passed away in 2020. But until then, I had an extremely cordial relation with him throughout my life. And although, like I told you, that publication was tough, never ever I had to have one, uneasy interaction with him. So thanks to him, and my luck also, I would say. And then after 2 years, I could get in Daniel Goldman's lab. And Daniel Goldman lab, when I go, I am the only postdoc, and other 2 PhD students who work on the retina regeneration are leaving because they completed. So then I kind of understood, thank God he did not interview me, then probably it would have been a no, and he did not interview me, and I started working. There were 4 goals he had in his R01 grant, and he gave me all the 4 goals, except one which he partially did. And first I thought, is it overwhelming? Then I said, no, you have no choice to choose. You are not in such a position. So I jumped in and grasped with both hands, and somehow it clicked and it worked out, and I could get around 8 publications in my 5 years of stay in reasonably good journals, and that's why I could fetch a job in IISER Mohali. So I will be presenting my research in IISER Mohali, and please feel free to stop me if I am exceeding the time. I will try to stick with the time. And so we have, this is the major theme of our lab, that is regeneration biology with a special focus on retina. We use 2 model organisms, one is zebrafish and another is axolotl, which I have started since 2019 because of my special interest on limb regeneration. I am not trained in limb regeneration or tail regeneration. I am trained only in retina regeneration. So I will explain some stories about the regeneration biology in general. So the talk will be broadly classified into these 4 sections. And so why retina regeneration? What is wrong with our retina, or what are the possible scenario in which our retina can give up? Many examples are there: retinitis pigmentosa, macular degeneration, cone rod dystrophy, retinal separation. It doesn't matter, but all retinal diseases in humans leads to irreversible blindness. So one statistical analysis done in US says US population, roughly around 5% of the population face some or the other retina related problem. Many a times pressure, blood pressure, and many a times it is diabetes. But what are the possible solutions? If it is a genetic defect is there, one could think of gene therapy or even stem cell therapy if you can replace the damaged cell, or even electrode transplantation which can sense light or no, there is some light intensity variation can be monitored to your optic nerve. But they are all not equivalent to vision. The ideal scenario will be regeneration of the retina from the resident population of cells. We know every organisms have the capacity to regenerate to some extent, or in other words, you are between extreme regeneration and no regeneration. Your life is in between that. If you have extreme regeneration, you are immortal. If you have no regeneration, you are mortal. So every day, like your skeleton, every 7th year, your skeleton is completely revamping. Like your skeleton's lifespan is roughly 7 years. If you are 70 years old, you are on the 10th version of your skeleton. So like that it is going on, and like that many other tissues also. The only complicated organ which has got some capacity to regenerate is our liver, but it is not true regeneration, it is compensatory growth. Simply by saying, if you have 5 fingers in your hand, I am chopping off your 3 fingers, but remaining 2 fingers becoming fat enough so that it mimics like a palm. I lost 3 fingers, they are not going to come back. But whereas these vertebrates such as fishes and frogs, if 5 fingers are there, 3 fingers are gone, 3 fingers of the same size will come back, and they are called epimorphic regeneration. So understanding the mechanisms of regeneration using animal models, many types of models are used: mammalian, avian, amphibian and fish models. Mammalian and avian are okay to use, but they have extreme limited capacity to regenerate the retina. So we use zebrafish as a model because it can reproduce year round, and it has external fertilization, transparent embryos, and they are amenable for microinjection and reverse and forward genetic screens. And also it shares human genomic, when you compare with human genome, it shares extensive genomic homology and synteny. One of our immunologists in Isar Mohali, he says, compared to mouse, zebrafish immune system is closer to human, he says. His name is Sharvan Sharawat. So he says, because of his own observation. And zebrafish can generate central nervous system neurons, because usually every organism have got least regenerative capacity for their central nervous system. So they can regenerate central nervous system very well. So this is a cartoon of the eye. So this is the cornea, this is the lens, and this is yellow color structure is the retina, and backside is the black colored pigment epithelium. Since it is cartoon, it is red in color. So if you look in ultrastructure, you can see the retina has got mainly rods and cones, which are photoreceptors, and then you have got horizontal, bipolar and amacrine cells in the inner nuclear layer, and then you have ganglion cell layer, which give rise to a axon which connects to your brain. And then you have a Muller glia, which is the only major portion of or major population of the glial cell. Normally glia is like an assistant for a neuron. In your brain it is around 1:3. For every neuron there are 3 glia. So glia basically is essential for the normal functioning of any neuron. So here the Muller glia normally provides lot of supportive function, and during regeneration this Muller glia reprogram, and it can reprogram and become stem cells. So what is the method we follow? We usually use a 30 gauge needle poke, a mechanical injury from the back of the eye, and so that the neighborhood region can act like a control. The same eye will give you the controlled region, and mechanical injury is going to be more closer to a natural injury rather than a chemical injury, rather than a light injury, because in nature animal may not expose to this extreme condition in a routine scenario. So evolutionary point of view, a mechanical injury is a better model according to my argument. Not going, slide. Okay, so this is a cartoon of the retina regeneration. This is an intact retina. This is outer nuclear layer where the photoreceptors are the inner nuclear layer, other neuron types are there, ganglion cell layer where the ganglion cell is there. If you make a stab wound, what happens? There will be a steady state release of a lot of cytokines, chemokines, etc. from the damaged tissues, which will be phagocytosed by this Muller glia, and they reprogram. And this reprogramming makes the Muller glia exhibiting stem cell like characteristics. Both Thomson factors and Yamanaka factors are expressed. All the 6 IPCs are expressed in these cells, and they migrate into various layers and it restores to normalcy. For this, so many genetic and epigenetic factors are contributing, because as the popular saying goes, winners don't do different things, they do things differently. These cells or these animals don't have new genes. They have the same genes as we have, but their way of expression and the way of functioning is different, or the timing is very important. For example, soon after injury in mouse, retina, the OCT4 levels are induced, which is an essential factor. However, within few hours, it comes back to the normalcy. Whereas in fish, induced OCT4 stays for a prolonged time until the reprogramming is completed. So I will not go into the detail. There are so many pathways that has been worked out, and this is the Muller glia that is allowing the reprogramming event. And there are so many factors that is like this is a tumor suppressor, and iron summon is a transcriptional repressor, and quite a few of the proto-oncogenic genes, and this is a proneural gene. So many of these pathways positively or negatively influence the maintenance of this progenitor. So the Muller glia has to become progenitor, and this progenitor has to give rise to Muller glia and also other retinal neurons. So this is all about the regeneration. So this is a transgenic line which we used, that is GFAP is a housekeeping gene, glial fibrillary acidic protein, which is a cytoskeletal protein which drives the GFP exclusively in the Muller glia. So you can easily isolate these cells, and as you can see in this window, it's a cell sorted, FACS cell sorting window, R3 window, you can see these are Muller glial cells. Whereas in control, this is another transgenic line which we have used, that is a tubulin, which is another cytoskeletal protein, tuba1 GFP line, which we say in an uninjured scenario, there is no expression of GFP in there. That means in an uninjured scenario, there is no need of reprogramming. Whereas if you injure it on 4th day, if you look, you can see a significant expression of this GFP driven by this tuba1 promoter, and which can be seen in this. And even if you give around 10 to 12 pokes per eye, you will get only 2% of the total retina responding to injury. So remember, around 40% of the total cells of the retina are Muller glia. So you get these cells, and this exhibits all the pluripotency factors. Hence what we did, we isolated these cells, we sorted these cells and delivered into the morula stage of a zebrafish. This is done not for any great reason. Whenever you submit a paper, the reviewers will say, don't use the word stem cell. They are against the word stem cell, although all the pluripotency factors are expressed. Then I took these cells and delivered into morula stage, and I saw, is it genuinely participating in embryogenesis? The answer is yes. It gives rise to not only neurons, it also gives rise to muscle, skin, and various other type of cells. Of course, it is a chimeric look. So I am personally convinced that it is acting as a embryonic stem cell, at least for a developmental biology point of view. So this is a typical injury site of the retina. The stab wound disrupts the retinal pigment epithelium, and this is the expression. This is a gene lin28, and my talk will be more on lin28 because it's one of the Yamanaka factors. And as you can see here, so in this box indicates subsequent pictures will have such kind of box. So imagine that it is a part of the whole retina, and retina is a huge cup-like structure. So lin28 usually meant for sweeping away or getting rid of let7 microRNA. Let7 microRNA makes sure that a differentiated neuron remains differentiated. If let7 is not there, then there are many spurious pro-proliferative genes will be expressed, and they will turn on to proliferation. So it's one of the important pluripotency factors, and it's also important, it's a marker of embryonic stem cells, etc. So it is coded in your genome in multiple places, and lin28 prevent the maturation of pri-let7 to pre-let7, that is the first stage of maturation, and then dicer converts into proper let7 microRNA. So lin28 degrades existing mature microRNA, and it will prevent the formation also. But one important thing to remember here is let7 prevent the translatability of lin28 also. So there is a kind of tug of war is there, who wins the race. So if let7 is there, lin28 protein will not be there, so there has to be some deviation of balance so that lin28 gets an upper hand. So this is a qPCR of this retinal progenitors at different time points post-injury, 0 hour, 15 hour, 2 day, 4 day, 6 day, and 8 day. By 8 day the proliferation is done. 4 days the peak of proliferation, that is maximum number of progenitors are induced after an injury. So you can see all pluripotency and Yamanaka factors are there, including ASCL1A, which is one of the major proneural gene, which is, I will not talk too much about that because it is an essential protein to convert a stem cell into neuron. And in regeneration you also see the glia back to stem cell, ASCL1A plays a major role. So you can see here, this is a western where we have fused the untranslated region of several of these genes as mentioned here into a bait protein, that is mCherry, and then increased the concentration of LET7. And you can see as the concentration of LET7 is increasing, there will be downregulation of this protein. And this work, detailed work, is published in one of my postdoctoral research in Professor Daniel Goldman's lab at University of Michigan. And so this is another story which I did in continuation, not exact continuation of this, but in principle, in connection with that, I started my research in Isar Mohali, and that work was published, and I am trying to tell about this story briefly. So many sonic hedgehog signaling pathways are important because it's a developmentally essential pathway, and we have confirmed their expression level, and we can see there is a time dependent induction. At 2 dpa the proliferation begins, 4 dpa peaks, and 7 dpa dwindles. And so we are kind of convinced that there is induction that is necessary, sonic hedgehog pathway is necessary for the regeneration to take place. Or the if you remove this is the induction of these genes in the proliferating population of cells. You can see SHH, patch, SMO, glia, these are all part of components of hedgehog signaling. And if you block it either by drug or by gene knockdown, you don't get proliferation or you don't get regeneration. So one such drug we used was cyclopamine, and cyclopamine can create a situation called cyclopia in zebrafish embryo. This concentration of around 30 micromolar we have used in the retina as well, because if you don't have the proper hedgehog signaling, you end up having a fused eye or fused facial structure. Sometimes human babies also born with this kind of defects. So you can see if you treat with cyclopamine, you don't get any regeneration. And so other method of knocking them down is deliver morpholino into the vitreous of the eye and then give an electric shock. You can see this morpholino will enter into the retina and you get effectively a gene knockdown. So if you knock it down with SHHA morpholino, you don't get proliferation, but you can rescue it with SHHA protein. So morpholino is there, but you can rescue it with protein. So this suggests that SHH is one of the, or the hedgehog signaling pathway, sonic hedgehog signaling pathway is one of the earlier events that is essential for regeneration to occur. And as I already showed you that SHHA is in the proliferating cell, if you look, the LET7 microRNA is selectively downward in them. So we just presumed, is it possible that lin28 expression and LET7 microRNA's downregulation in any way connected with the translatability of this SHH expression? So what we saw, SHHA, SHHB, these are two genes which is the paralogs of because zebrafish, all teleost fish have undergone a tetraploidization event around 340 million years ago. Because of this, they have got several paralogs. Like if your genome is 23 pairs, and what if, and you have total of 46 chromosomes. What if there is a tetraploidization event happened? Now your genome is 46 pairs instead of 23 pairs. So many of these pathway genes have got several of the LET7 binding sites, as you can see in red in color. And once you express this protein under GFP, and you can see in a dose dependent manner what we have done is take a GFP and also the microRNA binding sequence and transfect into cell lines and co-express with LET7 microRNA. And when you do Western blot assay, we have seen there is a dose dependent decrease in the translatability or the protein load of many of these proteins. So in general, you can say the sonic hedgehog signaling pathway is significantly regulated via the LET7 microRNA, and also this pathway in turn is regulating lin28 and ASCL1A. So there is a kind of feedback regulation that is going on between them. Now we'll move on to the second part of my story, which is also connected with lin28 and also histone deacetylase, which is a epigenome modifier. So this is what I am going to present is part of two story that has been published. So this what we have seen is the HDAC expression is adjacent to the proliferating cell, okay, and we wondered, it is induced but not exactly in the proliferating cell. When you look also, you can see HDAC expression is missing from this, and they are proliferating, but their very high level is present in the neighborhood cell. So now, if you see closely, you can also see that there is also a MYC and HDAC expression, and MYC and lin28 expression together, and in some cells you can see BRDU is there, but there is only the MYC, but there is no lin28. And when there is no lin28, that means the MYC is collaborating with HDAC and causing a repressive role. So MYC, being a proto-oncogene, can act as a repressor if it is collaborating with histone deacetylase, which is a transcriptional repressor. But the same MYC can activate lot of transcription factors if it is having a collaborator MAX. So we showed this more in detail by knocking down the HDAC, and HDAC's proliferation can negatively affect the proliferation even. So HDACs can also be negatively influenced by the transcriptional blockers such as TSA, which is some of the HDAC, many HDAC inhibitors are there. And we saw in this study that if you block transcriptionally or post-translational blocking of TSA, of HDAC using TSA, you can get a reduction in the lin28, and also you can also find that there is a concomitant increase in the qPCR level also, and this is reflected in the in situ also, and this increases the LET7 also. This is the main reason why there is a decline in the proliferation that occurs. But if you withdraw the drug, you can see that it is restoring back to normalcy. So based on this, we also did some study, which I will not show due to time constraint. So what I also saw in this, that lin28 also comes back when you withdraw the drug. So one thing we should understand, the regeneration is reversible. Even if you block it in one stage, after some time, if you withdraw the drug in a reversible manner, the regeneration ensues. That means the system is so plastic that it tries to restore the normalcy back. So I will not go into this phase because of time constraint. Yeah. If I have any question, I'll take it. Yeah. I'll quickly go to the acknowledgement slide, just 2 minutes. So this I wanted to show the immune angle of the regeneration, but since I don't have time, I will not show. So what we are trying to show is that there is a significant role that is played by the immune cells in in regulating the regeneration. So if not immune cells, you cannot get proper regeneration. So these are some other studies which we did from our lab. Even tumor suppressors have got a major role to play, and so I acknowledge because we have a fantastic group of students at IISER Mohali, and because of this was my first PhD student who graduated, Simran Kaur, and this is Saumitra, who is the second student, and Shivangi, who is the third student who graduated, and she is Mansi, who is the fourth student, and Bindya is the fifth student who graduated from the lab, and there are other 7 more students who are there in the lab, and we have lots of BSMS students, and it is very tough to keep them engaged in work. So because of that, we have to do lots of diverse areas of research, and also the funding agency IISER Mohali is a great funder for us, and STARS, ICMR, and DBT, CRB, and Wellcome Trust DBT India Alliance, and also our collaborators from within IISER Mohali, Anand Bachawat, Kuljit Singh Sandhu, Shashi, and Shravan Charawat, Mehak Sharma. They are great collaborators. Outside IISER Mohali, I had a collaboration with Professor Balasubramaniam and Professor Vinay Gupta of AIMS, and also Shilpi Minacha of IIT Delhi. So they are all great collaborators. And also I have included few of the pictures of my other MS students. They are all not there, and also I thank my family for being supportive. You can see. So I will take any questions if you have.
Okay, we will now open for questions from the audience to Dr. Rajesh, if anyone is.
Okay, I have a question.
Yes.
Why do you think humans do not have such regenerative capacities?
Very good question, and it's $1 million question. So the actual answer we can say is that there are many factors associated with that. So one of the major proposed mechanism is that HDAC which I was talking about, histone deacetylase levels and even your glucose levels in your body, glucose levels and histone deacetyl levels and also extracellular matrix levels, they can play a very major role. So the portion which I could not present, which is mainly talking about how HDACs are influencing the ECM buildup. So if you inhibit HDAC, there will be a tremendous buildup of ECM protein, especially the collagen. And if you get rid of that, there is a mouse paper which is published in Nature by Tom Ray in 2018, that if you block HDAC with the TSA, you can facilitate retina regeneration. Not complete, but it is far better. So what it tells you, that there is some ECM protein levels that is not optimal, that is present in mammalian system. And then other reason is that our immune system is very supreme compared to these animals. Like I was talking about axolotl. Axolotl have got a very primitive immune system. You can take the head of one axolotl and attach on the belly of another axolotl, it will happily grow. So it doesn't reject. So sometimes supreme immune system, even if you have a stem cell induction, it will immediately be cleared off. That is why delivering of stem cells in an injured area hasn't picked up the way it should have been. Because our immune system finds any even simple variation in the MHC level can attract an immune response and it will be cleared immediately. So, and these animals, especially waterborne animals, have tremendous capacity to evade pathogens. Their defensins and other, like crocodile serum has got, even to kill that famous hospital superbug, right? So it's a serum, will not let even that bacteria to grow on a plate. But it has around 50 or 60 different antibiotic, it has resistance. So these waterborne animals have got a great capacity to defend microbial infection, and regeneration is slow. So what we do priority is that immediately make a scar formation, wound healing and scar formation. You have a blood clot and regeneration is slow, and the priority is to prevent infection. You don't want infection. If you have infection, you don't have any answer to it. So we can live even if you don't have an appendage. These days people are using this spiny mice, which is having a better regenerative capacity than other regular mus musculus. So people are using it, but in general mammals don't have a good regenerative capacity except for nail, hair, maybe a little bit of skin, like that. Yes. Yes.
Thanks to the zebrafish model, it's been now possible for us to move on to humans. Gene therapy as well as stem cell therapy in human population, and regenerate the both the lenticular part and the retinal ganglion part. I think this is something that the collaboration that we had with him on zebrafish has taken us on to the humans. Today we must have taken care of about 2,000 such patients. Patients who come to LV Prasad for retinal regeneration and so on.
Great talk, Rajesh. Amazing. I'm going to ask you a philosophical question. Hopefully that will be motivating to some of the students here. 5 years of struggle, no publication, and then 5 years of amazing performance, and you get 8 publications out of that. There was this bridge 2 years in between when you're trying to get a postdoctoral position somewhere. What kept you going? What was the motivation? How could you stay sane?
Good question. It was a stressful period. Only hope was that I have to publish my thesis papers. So when I was defending my thesis, I had 0 papers. So if I leave with leaving them, they will never see sunlight. And if they don't see sunlight, then first of all injustice to the work, and second thing is my CV will be doomed. So I got one paper, and then subsequent ones I communicated with my boss and published in 2009 and 2010. But with that one paper I could get a position, and thanks to him not taking too seriously about my CV. So, but the remaining 5 years I can tell that, I was kind of lucky also. Otherwise it's not that easy and another good thing which I felt like doing was, when one project is around 40-50%, I start another project, so that when that will reach 70-80%, this will reach 30-40%, then I will start another one. So parallelly 3-4 used to go. So in that way every year 1 or 2 of my publications used to go, because I know age is against me. In India after 35 nobody wants you, and after 35 nobody wants you in anywhere. Yeah, so and he also said, Professor Goldman also said that, Rajesh, you can be there as long as I am here. And I know US has no retirement age. So then I felt, it is time to, same person, if I am not productive, he may say something else also. Although he is a fantastic human being to interact with, and I could see, and he was quite supportive. So that is why that 5 years, that again we don't know. While struggling this 2 years, you don't know that, whether 5 year will be good or not. So, but I could see this 2 year, at least I can get some paper. So that is what the only, the ray of hope that was running in the end of the tunnel. And that you badly need it, otherwise you can't go anywhere. So that is
sometimes success is just a matter of hanging in there.
Yeah, just hanging in there. Yeah.
Hello. Here. Yes. Okay. So it's a nice talk. So my question is regarding.
Okay. Yeah. Thanks.
Okay, so my question is regarding this HDAC inhibitors that you have used. So these are like non-specific deacetylase.
Yes.
So what you are trying to do eventually is either you are compacting the genome or either you are opening up the genome. How does it, like the opening up and the non-specific is leading to these kind of drastic phenotypes? So
you said the right question, and HDACs are not just meant for deacetylating histone, although their name says histone deacetylase. So we have done specific knockdowns also. Okay, I did not say because of the time. So normally you have to HDAC1 or whichever HDAC you are discussing, you have to knock them down and show as a parallel support. And histone deacetylase inhibition is also accepted by FDA as a strategy to curb tumor. Okay, so it is so just because it is an FDA approved strategy, normally it is okay for using that drug, but I agree that it can have non-specific effect. But one thing you understand, during reprogramming, Muller glia is a differentiated tissue, so it needs both suppression as well as activation of gene. Right, because it has to get rid of all the housekeeping gene and it also has to turn on. So when you block it, it is not that all genes will be turned on, although you keep the histone in an acetylated situation. Normally H3K27 acetylation level will be high when you block the, that is one of our yardstick to show that it may be blocking other acetylation events also, but we look H3K27 acetylation levels are going up because of HDAC inhibition. Accordingly you get a increase in transcription.
Okay, so like what I'm trying to ask the question is, like, does it kind of, once you give these kind of non-specific inhibitors, does it produce a statistical cells? Because now all the cells, there will be opening up of the genome, so there will be misfiring of multiple genes.
Yes.
And now you have a statistical set of cells which are expressing multiple things.
Yes.
The one cell which is having the correct set of the things, which picks up and then do all this kind of scenarios.
So normally how this regeneration happens, like you are making a hole, okay, so you have to walk the cells from the surrounding and they walk and fill the hole, okay, and then simultaneously while the proliferation is going on, it never reached its peak, it's differentiating also. So in the system it is a combination of events, and Neurons, whatever happens gene expression, they cannot reprogram because they don't have centrioles or anything, they cannot multiply. So if at all a cell has to respond, that has to be Muller glia in a regeneration context. But if you are putting like a TSA, if you put in the animal in TSA water, then it is often high doses will kill the animal also. So it's not a friendly drug at high concentration. So we put it into the eye, or inject into tissue, what we are talking about. Like I did not present about the fin regeneration and limb regeneration. So normally you can deliver, inject also into the tissue. So global change, what you said is correct. Global change, it may not be very specific. Yeah, but HDAC, it is very difficult to do a genome-wide knockout because animal won't survive. Because many regeneration-associated genes, you can't touch it during developmental biology. Every gene is important. So only option is you should have a conditional knockout, Krylloxp transgenic line, and conditionally ablate in the retina. So that is the only option. You can't get rid of in the developmental stage. Yes. Yes. Yes.
Hey Rajesh, marvelous work. Congratulations
Thanks.
to your group. So don't hate me for asking this. Kitish was a wonderful person, God bless his soul. But for a fish that already grows to 40 kilos, why did you want to make them bigger?
No, it is the rate. It's a very good question. It is the rate which we wanted to. So in normally in Indian major carp culture, what is the problem is first year growth is very low. It will grow only up to around 400 gram. Second year it will go to 2 kilo. That means first year only 400 gram. Next year it is going 1.6 kilo. But what happens, many of the ponds in any place, they are all not perennial ponds. They will dry off during summer. So our goal was, can we accelerate the growth rate up to marketable size? So 400 gram is, so that was the whole goal. Not the, and this 40 kilo, it comes in around 20 years. Not or so that even the dry ponds also can be used for fish farming. And he made it, he was maintaining also, we don't know whereabouts of that, he was maintaining somewhere those transgenic line. Unfortunately, DBT never had a policy on transgenic line, hence it's a mystery now, and he is no more. So, yeah.
Okay, I think there are no more questions.
We would like to extend our gratitude to Dr. Rajesh Ramachandran